Based word. Oh man. All right. Hi everybody. It's Dr. Simons here. Anyway, we're going to talk about eDNA Explorer, which is what I've been working on the last 2 years, a whole bunch of people, and how we can download the biosphere. Okay. So first off, I want to talk about something that you might have heard about over the years, especially if you watch the History Channel. But ecology has a Bigfoot problem. So you see, there's a lot of observations of Bigfoot. People have been reporting Bigfoot for centuries, and thanks to some geospatial information systems, or GIS, we can actually map out where people have reported seeing where Bigfoot has showed up. Now, I know what you're thinking, this looks like a population density map of humans, and you are wrong. Nobody lives up in Manitoba. All right, so what can we do in order to track down Bigfoot? And this has been a debate amongst Bigfoot specialists for a number of years. Step one usually involves going outside. That helps. There aren't a lot of hikikomori Bigfoot. Second is go and find a Sasquatch or Bigfoot, if you were. Take a photo. Now, I know what you're thinking. Those photos usually look blurry. Well, Bigfoot or big feet usually have the same ability as Clark Kent to like rapidly move and like make everything look blurry when you take a photo. Little known fact, especially also about Clark Kent. And then you also want to record when and where you took that photo. So this is where you can get the EXIF information or the GPS stamp on where you observe Bigfoot. And then here's the optional step, which is get additional evidence. That's really cool that you found a photo of Bigfoot. And maybe you even recorded when and where you got that photo of Bigfoot. But do you have any other data? All right. So it turns out that Bigfoot has been like, really good at not being found, which I know comes as a surprise, like, 7-foot ape human hybrid, and we haven't found it yet. But the truth is still out there. Anyway, this brings up an important point in ecology, which is like you can take photos of stuff and you can try to find species. You can try to track them by observation. We've been doing that for a long time. But lots of times there's species out there, not just big foots, that want to be avoided. There's lots of other things that either avoid humans because they're nocturnal, avoid humans because they know better, or they don't necessarily avoid humans, but they're likely to kill and eat you if you take a photo. You can watch Grizzly Man if you want to see an example of that. But mountain lions like that in LA are a good case. But yeah, there's lots of species that will not, for whatever reason, tend to avoid human contact. So photographic data is usually not very tractable. So you can do camera traps, but even still, there's a lot of stuff you're just not going to see photos of, especially of things that are really, really small, or in this case, would be very little feet. Okay. Luckily, life is disgusting. So one of the things we've observed over for thousands of years is that everything is constantly spraying or oozing or whatever. And it turns out that stuff is very rich in DNA. And this basically, this literal snail trail, so to speak, in the case of the snail, or the metaphorical snail trail in case of things that aren't snails, allows us potentially to start tracking where species are, even if we can't take a photo of them. So that brings me to the next point and what I've been working on now for years, which is, what is DNA? Well, good question, me. All right, so DNA stands for look it up. Okay, now DNA's got 4 base pairs, so adenine, thymine, cytosine, guanine. They are complementary paired to each other, so adenine and thymine always bind to each other. Cytosine and guanine always bind to each other. And for those of you with a computer science background, this is literally error-checked binary code. That is what life is. It uses these 4 nucleotides, although if you're RNA, you swap out the thymine for something called uracil, which is a fun fact. But yeah, it's just this very compact information storage mechanism. And it's also a structural mechanism because DNA is what codes for proteins. And everything is built around proteins. So this is, graphic design is my passion. I made this. But yeah, so this is what DNA is. It's just like these 4 letters over and over and over again. And from all of that comes all the emergent complexity you see in the ecosystem. So if you can figure out a way to like read this, you get down the road of like trying to figure out from the residue of everything that's around you, tracking species. All right. So this is where this whole idea of eDNA, and I was asked before the talk what the E stands for, and depending on the context, the E can stand for expensive. Although not so much anymore. We've gotten a lot better about like this. It's sort of following a Moore's law thing now with environmental DNA. Oh yeah, these are environmental. All right. Anyway, it's environmental DNA, which is DNA from the environment. And in case you're wondering, there's lots of other prefixes for DNA. There's also aDNA for ancient DNA. So DNA that you can sequence that goes back up to, well, actually, it looks like up to 3 million years now. I'm working with somebody on that. SED or SED DNA, which is just sedimentary DNA. All sorts of fun things. So anyway, wherever you can find DNA. But yeah, so we're extracting this from environmental samples and sequencing it and starting trying to figure out like what was there. So, and by the way, in case you're wondering, I have gotten a number of calls over the years, and same with my colleagues to try to get eDNA readings for Bigfoot. If you want to ask me that, any questions about that, like during the talk or after the talk, oh yeah, interrupt me by the way if you want. This is a conversation where the topic centers on me and my work. So it's my favorite kind of conversation, but you can interrupt me nonetheless. All right. So here's the steps for dealing with eDNA. So step 1, samples collected from the environment. Okay, so that's where the E comes from. So some of you who have gone out to the LA River have seen this before. You either, get stuff from the water, you filter the water, you get dirt and put it in tubes, and you can extract DNA from that. And you can also, by the way, get DNA from things like you can get it from ice cores in some cases. You can pull it from air filters. But the bulk of eDNA work is still being done with either water or sediment or dirt sampling. All right. Then DNA extraction. So this is the easy bake oven science aspect of what we do, which is you take your dirt and then there's like all these different steps to get everything out of there that's not living matter. So in the case of dirt, and then breaking the cell walls, loosening the DNA strands out of it, sequencing the DNA, like getting the DNA ready to actually run through the sequencer. There's just a ton of steps to it, depending what kind of material you're working on. But it's been done so much now. There are different specialized kits depending on the material you're working with. So there's literally like you can buy a box from like different types of boxes from labs that have all the materials and instructions to do things like, oh, you want to extract from soil, or you want to extract. There's even what was it? The company's called MoBio. They have one called the Power Fecal Kit, which I used a lot for a different project. So, that's for microbiome studies. But yeah, easy bake oven science, cool. All right, then you get all that DNA. Now you need to get enough of it that you can actually sequence it, because as you can imagine, like you're dealing with microscopic amounts of something, and you want to amplify it billions of times, and that's heat up a sample, you fill the sample with complementary nucleotides. So when you heat it up, the DNA unzips. And then when you fill it up with the rest of those amino acids. So the nucleotides, so A, T, C, and G, they do their complementary binding. So now your single strands become double strands again. Now you have 2 of them, you cool it down, then you heat it back up, and you keep repeating the cycle, and you have an exponential growth. Typically, you do this like 30 or 40 times. So you imagine like 2 to the 40. That's probably like gotta be like what, like 2,000? Anyway, so it's a lot of times you multiply it. So you have like large amounts of DNA to sequence. Okay. Then you could run through the sequencer, and there's different ways of sequencing, by the way. But the one I'm going to talk that we use a lot, and it's still sort of the workhorse of a lot of sequencing, is well, it's kind of like how it's sort of how this works. Yeah, so the workhorse of this for sequencing, this method is called Illumina. And the way it works is you take all your DNA strands and you get them to bind to like a treated glass plate. So you have all these DNA strands, now they're stuck here like shag carpet. And then what you do is you break the strands, now they're like single strand. And what you do is you wash a solution of treated base pairs. So in this case, like you have a treated base pair that's like going to be adenine, and then what you do is you have the adenine bound to, in this case, like you put a blue phosphor on it. And so what happens is you wash that solution over, the A will only bind to a T. And so when you wash it, it's only going to bind to that certain position as your let's say the first letter. You wash it and it'll bind, and it'll flash blue when it binds to it because the energy released. And so you can now record the position. By the way, this is an overhead camera. This animated GIF here is an animation of literally what this looks like zoomed in, but you're looking at billions of these different color flashes going on. And so you do this, you wash, let's say, a solution with A, then you clear it out, then you wash the solution with thymine, and let's say you has a red phosphor, so that'll flash red, and you keep washing and repeating the nucleotides with their phosphors, and they're going to flash different colors. And from that, you can rapidly read billions and billions of strands at once, just from the color position. Accuracy of this method, and usually I'm reading a couple hundred base pairs at a time, letters at a time, is about 99.99%. But you can get like ambiguous signals. So you can have stuff where like you can't tell if it's an A or T or an AT or C or whatever. There are what are called ambiguous nucleotide codes. And so like that's just kind of par for the course. You try to do error correction, but anyway. So you do this and you read up all these strands at once. Isn't that cool? Okay. So now you have all the DNA fragments. Now you have them all read. Now the trick is to figure out like what do these actually belong to. This is where you get to the big computer science problem. But you have all these different strands, and what we've had over the years, and this grows over time, is that we have all these different species out there that we've sequenced their genomes, or we've sequenced chunks of their genomes, and we know what the species is. And that all that information gets stored in a big central database. And there's a handful of these really big databases, which are basically they just store like, hey, here's like all the gene fragment, like here's like a and then you throw it against the wall, so to speak, and you see like, okay, like what for this fragment, what is the most closely matched in our database? And it's going to be like, let's say like what 300 like letters or whatever of your DNA fragment, what's the closest match? And it's going to, that method's going to say like, if you have really good read quality and it's something that we've sequenced and we know what it is, you can definitely get down to species level. If the sequencing has enough errors, or if it's something where like, this happens a lot with bacteria and fungi, where we have a lot of things we haven't like sequenced yet to make it like to put in the database. There's a lot of ambiguity, and all we can say is statistically like, well, I don't know what it is to species, but maybe I know what family it's in. And that happens a lot with like all the microscopic stuff where we're still like rapidly building up these databases. For the big multicellular stuff, that's a lot less the case now. Like, but there's far less plant and animal diversity than there is microbial and fungal diversity. Like, I mean, orders of magnitude more stuff for bacteria and fungi. So, hey. But anyway, this has pretty much always been the age of bacteria. Bacteria have been around for 4 billion years or so. Animals, maybe 800 million years old at the earliest, if you look at sponges. So we're relative newcomers. So 800 million years, not that long a time. Anyway, that's how we do that. We just throw it against the wall. Now, computation, as you can imagine, this is a very intensive procedure. There are ways of speeding this up. Okay. And then analysis. Okay, so once you got your species data and you have all the information like when and where did you get that species, like when and where did you find those samples, you can also go back and get environmental data. So this is a lot of stuff like once you actually get like, hey, I saw like these 3,000 species here and I was found at this location at this date. This is where we get into like all sorts of analysis, and this is where we get into this web platform that we've been building for the last 2 years called eDNA Explorer that help us do this. But there's a lot of different analytical techniques. So I'm going to go over the ones that are built into the website. And then later on, after, I'm going to get into other techniques where they're not built in the website because their computation is still way too intensive. But there are things that we can actually do with the data we generate with this platform. So going to go into like, how do we actually get environmental data associated with our samples? From our data, how can we tell what the prevalence is like for every species that we find in our data? Like how, like, let's say like how many samples are found in a project or how distributed they are, and then get into some other biodiversity metrics like alpha diversity, which is diversity at single location, beta diversity, which is how different locations are. And then we're going to talk about some comparisons with actually what are called traditional methods, which is essentially like going out there and taking a photo, let's say of your Bigfoot. And then we're going to get into some of the cooler techniques, which are way too computationally intensive still to like building a web platform, but like things like species distribution models, zeta diversity, which is the like we're going to 5 blades version of ecology, and then co-occurrence networks. All right. So first thing I'm going to talk about is like getting environmental data. So a lot of things in ecology, you want to get the context for if you have all this biodiversity, you want to start to look at like questions of what influences what species show up when and where. And that usually involves comparing it to environmental conditions at that time and place. In order to get that environmental data, people do take field data. So like, let's say if you're at a river site, you can get like river pH or other things like that. But there's a lot of environmental data that either it's difficult to get from in the field, or it just becomes really tedious to get in the field. And luckily now there's a lot of satellites that collect continuous data, basically so remote sensing data that we can go out and we can get all those map layers and then essentially take like, so this is like an example of a map stack, is we can download all these map layers and then say like, okay, take a point and like literally puncture a hole through all those map layers and extract all the environmental data at those locations. And now you can associate with your samples. Now you can say like, oh, what was the photosynthetic activity, or what was the elevation, or what, things like that at the point where I got all that biodiversity data. So that's something that we have built into eDNA Explorer. And I bet you're wondering, wow, who would manage all that kind of data? Well, it's our good friends Google. We're probably listening right now. Okay. So Google Earth Engine is the system that collates a lot of different map layer data sets. So generated by all sorts of different organizations. They collate it in a standard format so you can query it. So we actually have a whole set of scripts we wrote in Python and just basic like Unix bash scripting, where we can pull in hundreds of layers. We pull in about 250 out of like, I don't know how many thousands of layers Google Earth Engine has at this point. But we pull in about 250, and these are like various like socioeconomic layers. So things like we have like population density and we have it broken down by like age brackets. We have things like land use categories. We have a lot of environmental stuff like photosynthetic activity, artificial light at night. And then we have like climatic average data, all sorts of stuff like that. We pull that and we extract all that data from like the when and where of when you got those eDNA samples. And by the way, this is stuff that's all automatic and like all automated within like our eDNA Explorer pipeline that we've built. All right. So the next is like, we get that, and then here's some like basic biodiversity analysis, which is just literally like of all the DNA data I have and all the samples I have in a project. So let's say I have an expedition, I collect like 300 samples, okay? And you want to know like, well, what's really commonly found in my project? Like what percent of all the samples I have, what shows up and how often does it show up? It's just like real basic inventory stuff that a lot of, let's say, land managers who are some of our initial user base want to know, which is like what shows up, what's really common. And this is an example. So this is looking at, and this is, by the way, this is just a screenshot of the website. The website, by the way, now is live. We had our hard launch in the past week. And this is things like, okay, like this is from the LA River, the first 3 rounds, like the first 3 sampling rounds of the LA River, and things we found, animals. So we found a lot of these sort of stuff. By the way, most of the stuff we find, people get kind of bummed out because it's really boring because it's all these like tiny squishy things, like nematodes, flatworms, all that. Yeah. So a lot of that sort of stuff. But by the way, like it's a numbers game. Like these are really tiny. They're found everywhere. And then you get a ton of bacteria and archaea. And this is all the stuff that we coded on the platform. By the way, just to correctly get when you put in your data and to correctly pull out these like little icons, and the blue is the highlight is the common name or the most resolved common name, which by the way, shockingly for archaea is usually just archaea because people haven't come up with common names for these things. That was like 6 months of my life. And the reason is because there's like 4 million ways to do taxonomy and nobody agrees on it. I spent a lot of my time literally stitching databases together. So I learned a lot on my summer vacation. All right. So this is like one of the things we have built into it. So you can go in there, upload your sequence data and like when and where you got it, and we'll like provide this for you. So I think were really cool. Okay. Other stuff is like people want to know is like, okay, we have prevalence overall. We want to know, or let's say we want to know prevalence over time. So there's a lot of temporal trends, there's a lot of studies. Let's say with the LA River, we're doing this over months or years, and we want to know how much this fluctuates. So in this case, let's want to look at fungal diversity in the LA River, and we want to see like, okay, how much does this vary over time? And this is what you get. All right, so now if you want to go into like what does this all mean, that's way more to like read the tea leaves, but we're providing upfront on this platform the tea. And this is what you get. So we have all these, oh, look at those little icons. Doesn't that look like penicillin? Okay. But we get these temporal trends, and this gets to another thing you can do in ecology is look at over time. And especially if you're doing like deep time studies, you can see like long-term trends versus seasonal trends, all sorts of stuff like that. So this is something else we have built in, is to do stuff over time. We also have a lot of stuff to look at just by, we can look at even within things by site and by sample. So this is just saying like in each site, so this, in this case, the LA River study, this is things like Arroyo Seco, the Bowtie parcel. These are locations we've been sampling repeatedly. And this says like, oh, you found this species. What fraction of the samples in that site location did you actually find this? So this is like in a given location, because we usually get a lot of samples we collected to get a given site. And you want to know like, okay, how often does this show up? Because again, it's one of these things where like with eDNA, like maybe it was a fluke that you found this. Maybe it was just like something that happened to wash downstream at the very moment you sampled it. And so it's only going to show up like 1% of the time. And maybe it's just a fluke. Whereas if it shows up like every time, it probably is really there. Or it's probably like ecologically really there. So even like just because the DNA, just because the DNA says it's there, well, anyway. All right. Oh, now we got to start all over from the beginning. Okay. Okay. So other stuff, alpha diversity. Okay. Now alpha, first letter of Greek alphabet. What does that mean? Okay. So alpha diversity is just literally the richness of species in a given location. Whereas like it's some measure of the biodiversity at one location. And this is something we've like built into the site. Where you can play around with this. So, but anyway, anyone after the talk, if you want to like look at this, I can show you some projects where like this stuff is live. You can play around with this. Literally having gigabytes of data flying around, just so you can make these little violin charts. So this gives you an example of like you can compare this against environmental data. So a lot of things that people want to look at is like, well, how does like the diversity of a location vary against some environmental gradient? So in this case, this is an example I just did with like soil types. These are like, let's see, what soil types do we have today? These are haplochloropsis. Anyway, I don't know soil. But yeah, these are, let's see, torothents, xerophovents, xerophents. Anyway, people get PhDs in soil science, not me. But there's literally hundreds of these soil types, the USDA catalogs. And this is just a case, this is just an example I wanted to show, which is how does, let's say, fungal diversity vary between different soil types. So this is this is a basic environment, like ecological question, which is like, does the diversity of some category or organism vary a lot with some environmental condition? So this is an example with what's called categorical data. So soil type is not a great like a continuous variable. It is just literally like you have these X number of categories that we've made, and what do we get in terms of that diversity? And then the very bottom, I know the font here is really tiny, but basically we also do some statistical tests to tell you, does, let's say, biodiversity for, let's say, fungal communities, does it vary significantly between soil categories? And the answer in this case is yes. So that little thing down there that says p-value is 0.001. That's another way of saying that essentially there's like a 0.1% chance that these differences are due to some statistical fluctuation. So fungal diversity varies significantly between soil categories. Not earth-shattering results. They're actually boringly reassuring, which is good. Okay, now what does it look like we're dealing with continuous data? So this is another example. This is looking at animal diversity versus, in this case, this is artificial light at night brightness. So further to the right, the brighter it is. And this is another example. This is what it looks like when you do continual data. We actually like also fit the error bands for you. And we also do statistical tests on this. This actually, P is 0.13. This is saying like there's a 13% chance that the relationship is just due to some statistical fluctuation, which is not significant, or people usually do not consider that significant. So this is just saying like, at least for the LA River data that we have, that we're not dealing with large variations in animal diversity based on the amount of artificial light at night. That's partly also going to be due to the fact that everywhere we sample is pretty bright. So, ta-da. Wow, thanks, science. Okay. The next category we actually have built in is something called beta diversity. So beta diversity, I'm giving you like a year's worth of ecology. You should be thankful. Okay, so beta diversity is a comparison between two sites at a time. So it's not just saying like how many species, let's say, are in like one area. This is saying like, what is the similarity between, let's say, like any two randomly sampled sites you want to look at. And there's ways people can visualize this, but like you're usually dealing with large numbers of samples. And if you imagine like each sample may contain thousands of species, you're comparing something that has thousands of different degrees of freedom. That is thousands of like different, species categories versus some other things that have thousands of different species categories. And another way of saying that is it has thousands of degrees of dimensionality. So you're literally can generate a point cloud that effectively has like 5,000 dimensions to it or 10,000 dimensions to it. And so visualizing how much like your data, like how much all your samples are different from each other with beta diversity can sound like a completely intractable problem. And the way we make it manageable is we do what's called a principal component analysis, where we take that n-dimensional cloud and then picture a light bulb floating in n-dimensional space, right? And it's floating around and you're shining a shadow through that cloud and you're trying to angle the light such that it makes the cloud, the shadow of that cloud as spread out as possible. When you do that, you're literally getting what's called the first and second principal component, the greatest degree of variation. That shadow gives you sort of the two most important dimensions of your huge n-dimensional data set. So here's a toy example, like this is a 3-dimensional point cloud, and then here's a 2-dimensional shadow of it. And when we visualize beta diversity, this is kind of what we do. This is like a huge dimensionality reduction trick. And it usually, not usually, but oftentimes, even though you may have like incredibly large degrees of dimensionality, you can describe most of it with only like, let's say 3 or 4 degrees of variation. So this is looking at beta diversity this is bacterial diversity in the LA River. And this is saying like, okay, this is axis 1, axis 2. This is saying how much does it vary against this thing called the global human modification index, where like 1 is completely like modified, 0 is like untouched. That's color codes the points there. And this is a very large cloud. This is the 2-dimensional shadow of it. And this tells you that essentially like a lot of that variation can be reduced. This says like the first principal component, so like 1 dimension of that data set describes 36% of the variation in your data. The second most important one describes 16%. So basically with just 2 dimensions, even though you have this like 10,000 dimension data set, 2 of those dimensions describe like half of all the variation you're going to see in your data. And this says statistically that the bacterial diversity, the difference in bacterial diversity between your communities is statistically significantly different depending on like how modified it is by human activity. Again, boringly reassuring. You're manipulating, like if humans are manipulating the environment, they're usually manipulating like what bacteria show up when and where for all sorts of reasons. But this is a way of like reducing these very complex data sets to like a single picture that looks like somebody sneezed on a chart. But these are very common in ecology. All right. Now the question is, coming back to Bigfoot, or in our case, the capybara, which is a large rodent, which according to the Vatican counts as a fish for the purposes of Friday, which is another story you can ask me about after the talk. But is that, did you see it versus eDNA? And this is a thing that comes up all the time for good reason, because eDNA will let's say if you're dealing with a watershed, the eDNA could represent something that just washed downstream, and it could be 30 kilometers upstream and it washed down, you detected it. Versus, did someone ever actually see this organism? And so eDNA can move around a lot. And so it can tell you that something was at some point, but it might have been transported to where you are. And also because depending what material you're looking at, that DNA may be weeks old, months old, or like in really extreme cases, up to 3 million years old. And so comparing eDNA versus taking photos is an important question. And we actually have a lot of stuff built in to like do this as a sanity check. Some things are going to only be detected by eDNA ever, because no one's going to go out there and take photos of archaea or bacteria. And they're probably never going to get a photo of a mountain lion and survive. But there are a lot of things where we do get photos and we do get eDNA. So we actually have a comparator built in. We pull in all of our photographic data from the Global Biodiversity Information Facility, GBIF. And this is, by the way, a screenshot of their data set for the capybara. Oh, look how cute it is. So it's a giant rodent. People eat them. So this is, GBIF is based in Denmark. They are the sort of the global repository for the database of databases, so to speak, for like all this photographic biodiversity data. And we're working with them. And we actually compare our eDNA results to their database. They literally have billions and billions of observations stored in their database. So, and this is an example of like what we get from eDNA Explorer. So this is for, let's say, a local, this is just local to the LA River watershed. Here are all the fungal species we've identified, like that come up. Now local, by the way we define it is it's a half-degree box that we draw around all the points in a study. So let's take the Los Angeles area and then draw half a degree around it. It's actually relatively big. So GBIF has a ton of fungal species in that area, which is a relatively big area because it's like a good chunk of Southern California. And then the eDNA results are like what you're getting in the local LA watershed. And we actually show you like, well, what's in common between the two. So you're getting a ton of stuff in GBIF that doesn't show up in eDNA, partly just because you're dealing with a large area that you're drawing it around. And wait for that. And they're gone. Okay. So anyway, there's a lot of stuff that shows up because it's not only just a bigger area, but also when you're looking at GBIF, we're also just saying like, we're looking at the entire database, which means like every observation that's ever been made in that area. So, which can go back over 100 years. So this is just like, has anyone ever seen this in history? Has anyone ever seen this fungal species? Versus what did we see there at, this like 3-month period? So you can see though, like you can get a lot of biodiversity data very quickly though, because it's just like a handful of times. This is all over to assign certain confidence, like or comparison scores. So we actually have things that show up in both, which is really useful to know because it's a sanity check. Because we always get asked this question of like, well, how do you know if the eDNA says it's there? Is it really there? And we have this as a sanity check. All right. So that stuff that's all built in eDNA Explorer. You can go out there today and play around with it and make all sorts of great little charts. But we also provide the data to download to do even fancier analysis. As you can see here, this is fancy analysis. And I wanted to talk about some of the analytical techniques that people can use with the data that we're generating from eDNA Explorer that you can just dump out of the website and then do other stuff with. The first one, and we do a ton of this, but it's called species distribution models, which uses quote unquote machine learning. But the idea is that you can take your map where you have all your observations. So let's say where you got all your DNA data, and you look at all the environmental conditions where you got it. So you do this whole thing. So picture like you have all these like red dots here, like where you got all your DNA samples. And then you randomly do a bunch of sampling at points where you don't have any eDNA data, and you assume that you try to do this, you do this statistical assumption that says like, I'm going to randomly take points that are not where I got data and to say like those are absences or like background points. And you do a comparison of like, what did the environment look like where I didn't see stuff, and what did the environment look like where I did see stuff, and you build models to predict where different species will show up given your environmental conditions. So you can use this to say like where things are currently or where you expect them to be currently. And we're actually going to be using this. One of the things coming up is I'll be working with a group with eDNA Explorer to do this for all of California for fungal and invertebrate biodiversity. I know, which is what I've dreamed about since I was 5. But is to look, you can actually map out predicted biodiversity. So like we can look at what the diversity is at any given point we sample, but you're not going to exhaustively sample all of California. But what you can do is make predictions about biodiversity in places where you're not going to directly sample, which is very useful for a lot of reasons. You can also use these same models and then slot in future environmental conditions or predicted environmental conditions to say, where do you expect in 50 or 100 years for these species to show up? Which is, again, really important because you can do sort of things saying like, hey, if the planet gets 2 or 3 degrees Celsius warmer over the next 300 years, where do I expect these species to show up in the future? There's also stuff where like you can do current things today. Like, for example, if you're a wind farm and you want to estimate, like you want to do an environmental impact study, and you want to say like, hey, the species that are currently in the area we want to expand to, do we expect them to show up there or not? Can we safely expand this, or is it going to displace their habitat? You can also use these techniques to identify quote unquote indicator species. So those are species that are going to be sensitive to certain environmental conditions. Like you may have species that are really great indicators of air pollution levels, of soil salinity, and you can identify them. You can find your canaries in the coal mine using this technique. You can also identify like you may have hundreds of environmental factors you're recording, but maybe only 5 of them are really important for the most part, and to determining like where things are biodiverse or they aren't. All right, that is species distribution model. Wait, what? Oh, sorry.
How can you use AI to just do this for you?
Oh man, how can you use AI? Okay, somebody asked how can I use AI? Oh no, did I freeze?
Just the video.
Oh, just the video? Okay.
Well, is that like an idea that like there would be an AI kind of agent to this that's like an interface, and I as a simple user could just be like, I have dog poop in this area. Am I gonna, you know, like what's showing up here, and like what would show up here in 20 years, and how's that changing my soil for my garden?
So this is where I kind of hate the term like machine learning and AI, but these are the terms that get used. But is an example of a machine learning technique. And what you literally do is you take, let's say you have 50 environmental variables in your data set, and you want to use, and then you have like, hundreds of species in your data, and you want to predict like, well, where is each of those species likely to show up? So what you do is you take species 1, and you have all the locations where you saw it, and you have all these locations where you didn't. And you also know the environmental conditions where you saw it and where you didn't. And then what you do is you make a tree, and you make a tree where you fork. The first time you fork the branches is at your first environmental variable. Let's say it's, if you're dealing with aquatic species, let's say it's dissolved oxygen. And then you say like, okay, I'm going to fork that at dissolved oxygen. And then I'm going to next make a branch that forks at the next variable I want to look at, pH. And then the next one's going to be chlorophyll concentration. And what you do is you build this tree using like you randomly take 80% of your data, like 80% of the data where you saw it, like 80% where you didn't. And you make that as a training set, and you say like, okay, I'm going to randomly like vary the level at which those forks like branch. Like let's say if the pH has to be above, let's say I make the fork where pH is either above or below 7.5, and where dissolved oxygen is above or below 3 micrograms per liter or whatever. And you do that, and you say like, okay, I'm going to fork that each time. And if I were to drop a ball bearing down that pachinko machine, essentially, is it going to say, is that species there, or is it going to say it's not? And you see how accurate that is compared to like your actual data. And what you do is you build that model, and then you compare it to, then you feed in the other 20% of the data you didn't build the model with, and see how often does it correctly predict things being there or not being there. And you can evaluate the model accuracy. And then what you do is then you randomly subsample another 80%, repeat the process. And you can do this like hundreds or thousands of times. You aggregate, you make literally an average decision tree. This is some ways similar to how humans think. There's a lot of different techniques, and like you can build that model, you do it for every species. And this will then allow you to like, you literally just have this like average tree that when you drop the ball bearing down, it goes bing, 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 and then like more or less often goes in the correct spot. There are other techniques. Another one that gets used a lot is something called maximum entropy, which assumes that like your response, the response of some species showing up versus an environmental condition follows what's called a Poisson process. And the simplest way to describe a Poisson process is the probability of a rare event occurring. It was a statistical curve that was actually built to model the likelihood of cavalrymen in the Prussian military being kicked by a horse. It's a rare event, but you have enough data that you can model it, and it's like sort of this. So it has this like certain distribution curve, and you can like model things that way. Then there's all sorts of other things that like do something related to those decision trees, but like how they like build the trees or how they permute them varies. So there's random forest, there's gradient forest, deep forest is a band from the early 90s. But anyway, like that's the kind of stuff people do to do the species distribution modeling. And you can, yeah, so basically like picture, there's not really, I mean, I don't know. In marketing, you'll call it AI. In literature, they call it machine learning. And then at the end of the day, it's really just statistics. But anyway, but yeah, you could use this data. Like if you were to like map out dog poop and then map out environmental conditions where you saw dog poop, you could literally make a predicted heat map of where else dog poop is going to show up in LA and be relatively accurate. Yeah, I think so. Yeah, I've done this a ton with a couple different techniques. So, oh man, what else can we do with all this data? Here's something else that I'm really famous at, sadly. Zeta diversity. All right. So we talked about alpha and beta diversity. But what if you want essentially applying set theory to ecology, and allows you to do like nth order simultaneous comparisons between locations for biodiversity. And you're probably wondering like, who cares? Somebody played with the spirograph. So it turns out for weird reasons, this actually produces some interesting results, and it addresses some of the issues or limitations with alpha and beta diversity. So alpha diversity by definition will only tell you the diversity at one location at a time. And beta diversity can only compare two things at a time. And the problem is if you have like thousands of samples in your data and you want to try to like average that result, you end up getting really skewed towards the presence or loss of like really rare species. Because in picture, if you have 1,000 samples and some species shows up only once in one of them, it means that now when you compare the other 999 to that one anomaly, it skews the entire data set. And it's not very representative of what's probably really going on. So zeta diversity was partially developed to address that issue, because it says, okay, you can look at zeta sub 2, which is like the average overlap, like the average number of shared species between any two randomly sampled sites. You can look at zeta sub 3, which is the average overlap between any three, and so on. But as you go up to like higher orders of zeta, like zeta 10, zeta 20, you're looking at things that are held in common between 20 samples at a time. These are things that are really common. They're the subway sandwiches of your ecosystem. And so things that drive that gain or loss of things that drive changes in zeta 20 are probably things that are like your ecological baseload, things that are really common. And so it's really useful to have these like looking at different orders of zeta, because zeta 1 and 2 will tell you things that are changed by rare species, and zeta sub 20 or 30 will tell you things that are driven by common species, and allows you to make these comparisons simultaneously. But here's where it gets really weird and kind of interesting to me, is that you can also use this to address how ecosystems tend to assemble. Because it turns out, if you plot out, and here's an example of this, if you look at the number of sites, number of samples, and you go like 1, 2, 3, all the way out to 20, and you're plotting zeta sub whatever, sub n versus n, it's going to decay. And it turns out, so like zeta sub 2 is going to be smaller than zeta sub 1, zeta sub 3 is going to be smaller than zeta sub 2, and so on. And it turns out that it tends to decay, not all the times, but like 80-something% of the time, it'll either decay following a power law or an exponential decay. So these are two different mathematical trends. And if it tends to decay exponentially, it's evidence of what's called a stochastic ecological assembly profile. That is, what species show up in your ecosystem is kind of a random process. It's your castaway, your shipwreck model. It's whatever showed up, now there's your ecosystem. If it tends to decay via power law, it's evidence of niche differentiation, and niche differentiation is this idea in ecology that if species A is already there, it will influence the likelihood of species B taking root if it shows up. So because species A and B may have similar ecological niches, and like it may either exclude or it may actually be complementary, but they are dependent on each other. And so that'll tell you, like, those two different decay curves will tell you essentially like, oh, this ecosystem is either driven by this mechanism or this mechanism, literally just by like looking at this math decay, which is weird. It's like it's almost like just playing the numerology with us, but it bears out like time after time after time. We've looked at this a lot of ecosystems.
So the more interdependent one was the power law distribution?
Yeah, power law distribution. And on top of that, one of the things that's kind of interesting is ecosystems that tend to get disturbed tend to have these community profiles that look exponential, or like they tend to be stochastic. Basically, the more you disturb an ecosystem, the more it looks like a random process. And so like ecosystems that normally under undisturbed conditions look like they're niche differentiated will tend to shift over to being stochastic. That is, they tend to go from power law to exponential. So literally, I can look at just this decay curve. I can tell you, even without knowing what species are there, I can tell you that there's evidence. But at the end of the day, like, life is limited by thermodynamics, and thermodynamics is basically stats. So there's probably something going on there. All right. Zeta diversity. Wow, look at that squiggly. Okay. One of the other techniques that people use with all this kind of data, and this is really useful for ecology, is co-occurrence networks. So with co-occurrence networks, what we're doing is we're actually looking at, if I see species A, how likely am I to see species B? If I see species C, how often am I likely to see A or B? And basically what you're doing is you're making the social graph, so to speak, of the species in your ecosystem. So that is like, if you have thousands of species, you can, if they're likely to show up together, you can draw like a link between them. And like the more likely they are to show up together, the thicker you make the line. And you can literally make these really dense networks where you map out like how species tend to co-occur, or how they often, or how they may like actually repel each other, like counter co-occur, so to speak. And you can make these networks, and you actually look at like, oh, if this species shows up, is this one likely to show up, or is it likely to actually be repelled? And it allows you to start inferring all sorts of ecological patterns. You can actually use it to like guess at potential food webs, other ecological interactions, things like pollination networks, even things like what are called ecotypes. That is, like, if this species tends to show up all the time with this species, it could be because they're interacting somehow, but it actually could be just because they all have a similar ecological niche, and they just tend to always show up because they all both tend to like this like one particular environmental condition. So it doesn't necessarily address these questions directly, but allows you to start very quickly like winnowing down like a lot of different potential hypotheses for what's going on in a very complex system. Because at the end of the day, we are dealing with very complex systems. We're dealing with terabytes of DNA data that get distilled down to like millions and millions of lines of like, this species showed up here at this time. Millions or billions of components, and distilling it down to something that you can write in an index card, give it to a land manager and say, you need to dim your lights this much. It's causing these shorebirds to not nest. And there's actually a personal experience of mine, is spending 1.5 years of my life to do the field work and the computational modeling and the writing to literally hand 2 numbers to the state of California and say, like, yeah, don't get brighter than this. So those are 2 very important numbers. All right. Okay. So where's this all going? So what are we doing next with eDNA Explorer? So right now we deal with just with what's called meta-barcoding data, which is those like short couple hundred base pair fragments of DNA, which kind of just get like look at like large branches of the tree of life. But there's 2 other types of genetic data that we want to incorporate. One is called qPCR. The Q stands for quantitative. So qPCR and then metagenomics. All right. So qPCR data, and I'm actually currently working on this right now, is similar to the same sample collection mechanism as getting meta-barcoding data, but the difference is as opposed to just like sequencing and throwing everything against the wall, what we did with meta-barcoding. In this case, we actually design a very specific what are called primer, that we actually look for a very specific, we design it for a species in mind. So we're designing like these 2 little fragments of DNA that'll clip out one particular fragment of DNA from your sample that only belongs to like a species. So it's targeted generally to a single species. So you will use qPCR to clip out one particular kind of species fragment of DNA, and then you amplify it a ton of times. And the way you say that it's there now, this is where the quantitative comes in, is that you attach a dye to your DNA fragments, and as it keeps doubling, you say, all right, if I keep doubling after, let's say, 25 cycles and it's brighter than this level, I'll say that species was there. And if it's not, I say it's not. And so it's just basically a way of saying you try to do single species targeting for your biodiverse data. So we're building this right now into our pipeline. So I'm doing this currently so that people can do a lot of things saying like, hey, I want to look at, and this comes up a lot really with fisheries. It's like we're removing this dam. We want to see if the salmon are reintroduced to this river. I don't care about any of these other species. I just want to know, are the salmon there? So somebody designs a salmon-specific primer to only look for this like salmon DNA. They do this, they amplify the samples, and they go like, okay, it seems in 70% of the samples, the salmon show up. Okay, they're probably there. So that's something that we're building into eDNA Explorer currently. And it's got a lot of similarity with metabarcoding in terms of like computational stuff. It's a lot less computational overhead than the metabarcoding because you're dealing with like one species as opposed to like 40,000 or whatever. All right. The next technique is metagenomics. This gets gnarly. So metabarcoding, you're only looking for like usually one particular gene for organisms. So you're looking like some common gene that they share. Metagenomics is literally everything at once. It's throwing everything in the wood chipper. Every bit of DNA you can get, throw it in there, sequence all of it. And then when you sequence all of it, you want to try to then start to reassemble it into what are called like metagenomic assembled genomes. So you're basically taking like all these crazy fragments, I mean, billions of these fragments, and then trying to figure out like, okay, do all these fragments belong to the same species? And do all these fragments belong to a different species? And if so, like, can I reassemble their genomes? And computation, the way this is done is picture if you take a bunch of books in a library, throw them in a wood chipper, and from these like fortune cookie length sentences, you could actually reassemble the books. And the way you do that is you get a fragment and you look at like, hey, is there any likely overlap in any region of this fragment to this fragment? If there is, you now can stitch them together and make a longer fragment. And you just keep repeating that process until you get like what you think is probably at least the largest chunk of the genome of that organism. And you can do that with a lot of them. And you can say like, okay, these are like my 30,000 putative species in my data. And so you can imagine like there's a good degree of reliability. And like we do this with like genome assembly. This is like in a sense how the human genome was assembled, but the difference was we were only collecting human genetic material. This is like reassembling the human genome when only 1% of your DNA is human. And so you can do this and like try to like statistically infer like, well, this probably belongs to this, and these things are separate. And you're probably wondering, well, how do you know this big chunk of DNA really belongs to this bacteria, not to this bacteria? And that gets into some other statistical inference models. And this is gonna be a little hand wavy, but this gets really tricky. But one method that gets used a lot is something called a Kamer frequency distribution that literally looks at how often do certain combinations of genetic letters show up. And those combinations, depending on like what length you're looking at, are distinct between species. And again, it's a numerology thing where you don't even know what they are. You can just tell like literally by looking at the graph of like how often are these different fragments shared, are these different patterns shared, that this belongs to this species, this belongs to this one. So you can start to peel apart and say like, I don't know what these are yet. I just know that they're different. And as you can imagine, like we haven't built this at all yet into our platform because computationally, this is a lot more work than metabarcoding and a lot more work than qPCR. But as computational power gets cheaper and cheaper and cheaper, this is eventually where we start going because this allows us to do some really crazy things. Because unlike with metabarcoding, which only can go down to species, this allows you potentially to go down to individuals. So not only a turtle, but which turtle? And of course, now you're probably wondering, like, doesn't this sound like Gattaca, where now I know which individuals it is? And the answer is yes. So you can start doing like individuals of a species. You can look at, like basically you start looking at how genetic diversity within species will change over time. You can start watching evolution happen in real time in your data. career. With that, though, I'd like to say there's no I in team. And I want to thank all the people I've been working with over the last 2 years on this. So eDNA Explorer, again, we are now live. I want to thank the people, the Meyer Lab at UC Santa Cruz I've been working with, the sliced bread design team, so all those like graphics and everything. This is the front end developers I've been working with. That includes Sumo Creations and people that help support all the funding for all the sampling we're doing in the LA River, which is the LA County Board of Supervisors. And with that, questions. Woo!
With qPCR, are you able to dive deeper into like population numbers too?
So the question was like, with qPCR, can you dive into population numbers? So like, you mean like, very like looking at differences between individuals, or
Having better estimates of like not just is this species here or not, but seeing like is the species plentiful or not?
Oh, okay. So you're like abundance first. Okay, so can you use it to get abundance? The answer to that, there are projects out there I've seen, like people have done like various techniques to try to estimate, like especially like let's say in fish, to estimate abundance from like eDNA concentrations. It's the like because you're dealing with like all sorts of like material transport of DNA and like what's the degradation rate, what's the transport rate of like how that DNA is flying around.
Sampling sites.
Sampling, yeah, it's like I've seen models where people can kind of like get like a reasonable guess at it, but it's like a lot of work and a lot of modeling. And at the end of the day, you're getting something that's like sort of hand wavy for a single species. Maybe one day we get a better handle on like eDNA concentration versus abundance, but I don't know if we ever will. I personally only work with presence absence. I work with presence absence, and I can at least, I would go the route of, if you want to try to get estimates of abundance, I would actually go the route of do a bunch of sampling and just see the prevalence of it, like how often does it show up. It's probably it's probably more reliable than trying to go like from concentration of DNA to abundance. Because also you're dealing with different shed rates, and especially where you're dealing with like body mass, it gets, there's just too many parameters in the model.
7 badgers
could do it.
Well,
If you already know the diversity of the species.
If you counted all the individuals.
So this is okay. So the question is, could you do this metagenomics? The answer is yes, and that currently would be a computational moonshot. In 10 years, it will not be.
Yeah.
But what you could do is you can do what's called population genetics, where you just say, if you can get like individual level genetic diversity, so now you have like how many individuals are at this site, and you look at the genetic difference between those individuals, and then there's a thing in population genetics where you can look at genetic diversity and population size to like estimate like effective population size. That might be a technique, but like that's one of those things where like we can on paper say like, yeah, you could do this. Computationally, probably not for like another 10 years is that gonna be realistic, but that we are gonna get there. But it'll get, but it's that's like one of those, that would be like saying in 1950, can you get to the moon? The answer is like probably. We know technically how we can do it, but we haven't done it yet. And this is just like, okay, give Moore's law another 10 years, we'll get there.
Sweet.
Yeah. Is that what you're trying to do?
No, I'm just curious.
Oh, okay.
But eventually that'd be cool to do, but not any of my current projects.
Yeah. So, all right. Any questions out there in internet land? Oh, man, they're all
yeah, I had a question. I think you talked about spinning up a B Corp related to this. How's that going?
As of 2 days ago, we are now incorporated.
Cool.
Yeah. So eDNA Explorer is now, we're now incorporated.
Capitalism how?
Yeah, capitalism's cool. I own the means of production. I am the means of production. Or at least some of it. I code a lot. So, but yeah, this is an actual functional website. So if you go to eDNAexplorer.org, you can play around with it. So. Thanks, audience.
Yay. Yeah, your video cutting. Got anything else?
No, I think I can stop sharing.
Say goodbye, Levi.
Bye.